Okay, we're going to go ahead and get started. Firstly, hello and welcome everyone to the webinar. We're very excited to have you here today with us. My name is Kayla Ripple, and I'm a principal associate with the Lenfest Ocean Program. I'm joined today by my other Lenfest colleagues, Emily Knight and Victoria Bell, who will be dropping links to resources in the chat for you. For those of you who are less familiar with the work we do, the Lenfest Ocean Program is a grant-making program that funds ocean and coastal research projects to address needs that are facing decision makers and stakeholders. You can learn more about us and the projects we fund at lenfestocean.org. And while you're there, you can also sign up for our newsletter. And through the newsletter, you can receive updates on projects like the one you'll hear about today. Today is the last webinar in our Climate Resilient Fisheries webinar series, a series that highlights projects from our Climate Resilient Fisheries grant portfolio, where we are supporting projects that bring together scientists, managers, and stakeholders to better understand how changing climate and ocean conditions will impact fisheries in the US and globally. Our hope is that the results from these projects can help inform conversations around fisheries management so that fish stocks and communities are more resilient to future changes. And if you're interested in learning more about those projects, Emily is also dropping a link in the chat with links to each of them. For today's webinar, we're very excited to have joining us several members of a research team that's focused on understanding the role of habitat and life history in fisheries vulnerable to climate change. And Emily's dropping a link to their project page as well. Right now, the research team is working to finalize and publish their results, but you will hear some of their preliminary insights and findings here today. So Dr. James Morley with East Carolina University and Dr. Verena Wang, who is currently with the Gulf of Mexico Fishery Management Council, but conducted this work while she was with East Carolina University, will be presenting their results today. And other research team members are joining us along with Verena and Jim, including Lorenzo Cianelli, Marissa Baskett, Mark Nelson, Matt Ajamian, and Stephanie Archer. They'll join us at the end for Q&A. And before we get started, just a few logistical things. To prevent any feedback or echoes, all attendees are muted, but we do want to hear from you. So please feel free to drop your comments or other resources on the topic that you'd like to share in the chat box, and type your questions in the Q&A box. I'll be keeping track of those throughout the webinar, and at the end, I'll read the questions aloud for the research team to answer. If we don't get to your question, or if you have follow-up questions, please feel free to reach out to us or to the research team after the webinar. And lastly, this webinar is being recorded. We'll distribute the link broadly afterwards, so please feel free to share that with others who may not have been able to join today or didn't hear about it and might be interested. And with that, I'll turn it over to Jim to get us started. Let me stop sharing my screen.
Here we go. Or I got to share my screen. Yeah, thanks for that introduction, Kayla. And thanks to you and Emily and Victoria for putting this webinar together, and appreciate everybody for showing up. I'm sure many of you are snowed in at home like I am. Yeah, this is our team. I think Kayla introduced everybody, so I'll move right along. So, as Kayla said, our group is part of this cluster of projects that we're all focused on different aspects of understanding how climate change are interacting with fisheries and marine species. Most of you, probably all of you, are aware that one of the big responses that marine species are having, get rid of this, to climate change are these big range shifts. And this is an example with silver hake from the NOAA's DISMAP tool. You can see on the right there that silver hake have shifted northward over the last 4 decades, about a degree and a half of latitude. And then looking at the 2 snapshot years, it suggests that this shift has been due to an increase in abundance, maybe an increase in productivity in the Gulf of Maine, whereas the southern area looks a little bit more unchanged. So our group came together with this shared interest in trying to understand the mechanisms behind species climate change responses, including distribution shifts, but other responses as well, and to better understand the mechanisms behind what drives climate change responses. So certainly understanding the mechanisms better and the ecology that shapes these species responses is a more effective way for adaptive climate management, climate adaptive management. Of interest to our group was that climate change responses vary among species. So some species seem to be responding really rapidly or really sensitive to changes in climate, while other species don't appear to change as much. So we were curious about that. And also, it's noteworthy that a lot of previous distribution analyses have been conducted at the species level. So by aggregating all different life stages of species together to look at species responses. And so there's potentially a lot of valuable information lost with that types of approach. And that leads to our question is can spatial distribution patterns across the different life history stages of a species help identify species vulnerability to climate? And that question certainly feeds into NOAA's goals of their fisheries climate science strategy on multiple levels, understanding mechanisms change, projecting future conditions, and tracking and looking for early warnings. So species require multiple habitats to complete their life cycle. And this is exemplified here in this figure in one of Lorenzo Schinelli's papers. He's part of our team, using walleye pollock in the Bering Sea. You can see each map is showing a different life stage for walleye pollock, ranging from eggs and larvae up in the top left to the older life stages on the bottom right. And you can see the spatial patterns across these different life stages are quite different. And some habitats that are required by different life stages are shaped by more dynamic environmental variables, things like temperature or salinity, whereas other habitat requirements for different life stages are much more fixed in space. And, the classic example of that would be natal homing of adult fish, Pacific salmon, other anadromous fish that return back to their natal stream for spawning. Although, natal homing is more widespread beyond just anadromous fishes. But, these these spatial constraints of habitat that are fixed in space aren't just restricted to examples of natal homing. On the continental shelf, there's very distinct habitat features that are fixed in space. And as an example, I'm showing here this benthic map of the Northeast US shelf, which is showing different distinct habitat types based on sediment and based on depth. And, certain species and life history stages of species might respond to and really rely on some of these discrete habitat features on the continental shelf. And this, also can include important coastal features such as really productive large estuaries in terms of the East Coast. Major estuaries like Pamlico Sound, Chesapeake Bay, Delaware Bay might be really important for particular life stages of species on the Atlantic coast. So a useful statistical tool, an approach had previously been developed by Lorenzo Schinelli and others to quantify this use of space over a species' life history. And they're using this term spatial constraint, where here is defined as a small portion of the species range gets consistently used over time, over years by a particular life history stage. And so a higher value of spatial constraint would indicate that that particular life stage, over many years is really tied to that geographic location. And then vice Vice versa, a low spatial constraint value would suggest a life history stage that's more dynamic from year to year. And previously, they applied this statistical approach to several subarctic ecosystems, not just in the United States, with a number of groundfish stocks. And this figure kind of synthesizes the big results from that recent paper. And they showed that spatial constraint decreases with ontogeny. So you can see on the left side of this figure, we're looking at early life stages of species, and there's a greater likelihood of higher spatial constraint for those early life stages, and a lower constraint for adults and older life stages. But perhaps most interestingly, there was a lot of variation among the different species there, and that suggests that there's potential to use this tool to, compare different species in their, their level of constraint across life histories, that this might be a valuable tool to assess this sort of thing. And we're also curious if species life history traits correlate with this variability among species. So in a second, I'm going to turn over to Verena Wang. She was the postdoc that did all the hard work on this project, and she's going to share results, and she'll go through the process, the statistical process, with a little bit more detail. Specifically, our goal for this project was to extend this approach to more species and into different types of ecosystems, including temperate ecosystems, and also to relate the variation we see among species in their level of constraint to other life history traits. And the hypothesis there is that's kind of what we came into this analysis. That's what we were expecting, that species with more complex reproductive ecology and more varied habitat requirements across their life cycle will exhibit greater amounts of this geographic constraint. And ultimately our goal was to inform management and provide another tool for the toolbox for management to use. And so after Verena's done going over the results of the paper, the forthcoming paper, I'll return and share some parting thoughts. So Verena, are you ready to take over?
I am ready. Can everybody hear me?
I can hear you.
All right, great. Thanks, Jim. Next slide, please. So I'm going to pick it up from here and talk about our spatial constraint analyses and explain spatial constraint in a little bit more detail. But before I do that, I wanted to give a quick data acknowledgement. All the work that I'm going to be talking about today is based on long-term fisheries independent surveys, and I just wanted to acknowledge the folks who make these surveys happen, as well as the people at the Science Center and at BIMS who facilitated access to this trove of super important data for us. Next slide, please. So what is constraint? We're all familiar with the concept of environmental constraint. It's really what we're thinking about when we talk about species range shifts. A species has a particular environmental tolerance that limits where it can live, and it can move to follow that environmental envelope as the climate changes. That's an environmental constraint. When we talk instead about a spatial constraint, now we're thinking more about a geographic anchoring. So to illustrate the difference between these two, if we start on the panel on the left, the fish in that left panel is pretty happy to be living in its preferred light blue environment. In the panel in the middle, the fish is experiencing a change in conditions, in environmental conditions, and it shifts its location to stay within its preferred light blue environment. That's an environmental constraint. But when we look now at the panel on the right, this fish is experiencing the exact same change in environmental conditions as the guy in the middle, but it's not able to move to its preferred light blue environment. This fish is stuck in a suboptimal environment because it has an affinity for its little house here. This fish is spatially constrained, and it's spatially constrained because it has a habitat preference that's anchored in space. And a spatial constraint like this can result from anything from a limited nursery habitat to a really specific bottom feature that it requires to needing to return to the exact same spawning ground all the time. These are all habitat requirements that could result in spatial constraint. And spatial constraints can occur at any life stage and can be different across life stages within the same species, as Jim mentioned before, because as we all know, marine fish can use vastly different habitats to complete their life cycle. Next slide, please. So now that we know a little bit more about what spatial constraint is, I'm going to walk us through how we model spatial constraint. I'm going to do that first conceptually here, and then in a tiny bit more detail on the next couple of slides. So spatial constraint is made up of two components, consistency and extent. Consistency is really a measure of how often the same spatial habitat is used by a life stage year after year. And extent is the proportion of the area that's occupied by that life stage relative to the total population range. So in this example here, we have two life stages, juveniles on the top row and adults on the bottom row across a series of years from left to right. And if we look first at the juveniles, the blue square here represents the total population range, and the red circle shows the core of the distribution of the juveniles. And so the juveniles here have a pretty large distribution. That red is big relative to the blue, and the area of the distribution moves around, and it's in a different place year after year. So this combination of a pretty widespread spatial extent combined with a low degree of spatial consistency results in a low degree of spatial constraint. So in this hypothetical scenario here, the juveniles are going to be able to move around and shift ranges easily to follow an environmental preference because they can kind of be anywhere. They don't have a place in space that they need to be. If we look at the bottom row with the adults here, the adults are a different story. So the adults here have a really limited distribution. That red circle is really small relative to the blue, and it's always in the exact same place every year. So this combination of a really high degree of spatial consistency combined with a really narrow spatial extent results in a really high degree of spatial constraint. So in this scenario, the adults are going to have a hard time shifting to follow a change in environmental conditions because they clearly have a place in space that they need to be. Next slide, please. Okay, so the two components of spatial constraint, consistency and extent, are both modeled using a GAM approach. And this is using a technique that was developed by Cinelli et al., which Jim mentioned earlier. And consistency is really, again, an estimate of repeat spatial habitat use. And it's estimated as the difference between two abundance models, one that includes location and one that does not. So we use a base model that looks at abundance while factoring in year and also the day of the year to account for phenology, and then a habitat model that's the same as the base model, but also adds latitude and longitude coordinates on top of that. So the ratio of the error between these two models gives us an estimate of basically how important space is and estimates the consistency of that spatial habitat use. And just to kind of quickly visualize this, I'm showing you an example here of summer flounder on the Northeast Shelf, and these are just adults during the spring season. And so the big map on the left just shows you the total footprint of the sampling area on the Northeast Shelf. And the grid of figures, the bigger figure in the middle is summer flounder abundance. So those heat map colors are abundance. Again, on the Northeast Shelf, and that's across each of our sampling years. So if you look at some of the years near the top, you can see that summer flounder are kind of at the edge of the shelf in southern New England and in the mid-Atlantic bight. That's where those brighter colors are. But if you look at some different years, so maybe towards the bottom, summer flounder are kind of more spread out and they occupy more of the shelf area. So their spatial habitat usage is not consistent. They're kind of in a different place and not always in the same location every year. Next slide. The second component of constraint is spatial extent. And extent is modeled using, again, a GAM approach, this time using a delta model or a two-stage model that accounts for both probability of occurrence and abundance, as well as spatial coordinates. So if we start with the figure on the left, the big figure on the left, this is summer flounder again, and the green area is again, the total sampling area on the Northeast shelf of these surveys. And the black dots are a standardized grid of summer flounder occupancy. So basically anywhere that a summer flounder has been caught at any size and at any point throughout the history of the survey time series. So this represents the total occupied habitat. And we can see that summer flounder, based on those black dots, are found kind of over most of the Northeast shelf, but they're kind of absent from the Gulf of Maine. And so next in the panel in the middle, what we did was we projected our modeled abundance for summer flounder for just the adults in the spring onto that occupied habitat grid from the left panel. And finally, from this modeled abundance, we move to the panel on the right, where the red dots here represent the area that encompasses the core of the distribution. So it's where 75% of the abundance from that middle figure is located. And so spatial extent is basically what percentage of the total occupied range is the core of the distribution. What percentage does that take up? So these two scores, consistency and extent, are combined together to form constraint. And all of these are dimensionless values that just range as scores from 0 to 1. Next slide, please. So Chinelli et al., like Jim mentioned before, applied the spatial constraint approach to subarctic fish stocks. And we applied it here to 2 temperate marine ecosystems, the US West Coast on the left side here, and also the US Northeast shelf on the right. And for the West Coast, which ranges in the south from California through Washington State, sampling was based on 16 years of groundfish bottom trawl data. And on the West Coast, there isn't an ichthyoplankton survey that covers the entire coast. So we used pre-recruit data as our early life history stage. And that data is from 2 midwater trawl surveys that target pre-recruits. In the Northeast, on the right side, the Northeast ranges from Cape Hatteras in the south to the Gulf of Maine in the north. And our analyses here are based on 30 years of groundfish bottom trawl data. And we included a nearshore survey also to extend our coverage, and also ichthyoplankton data from both the Ecomon and the MARMAP surveys, which do cover the entire shelf region. And I should also mention here that in the Northeast, we did model 2 seasons, the fall and the spring, separately. And on the West Coast, we ended up analyzing everything combined into a single season because there are fewer survey months and also because there's a less distinct seasonality. Next slide. So in total, we modeled spatial habitat constraint for 29 species on the West Coast and 23 species in the Northeast. And for each species, we applied this spatial constraint modeling to 6 size groups separately, plus an early life history stage when that data was available. And I know there are a ton of species listed like really close together here, but I just wanted to give you guys a sense of kind of who the players are in each region. So the species that we chose were chosen because they were really well represented in the groundfish surveys, and they also represented kind of a span of different species types, a span of different kind of broad life history groups, and that includes some flatfish, elasmobranchs, gadids, a few pelagic species, and then some rockfish on the West Coast. Next slide. Okay, so what I'm going to do now is walk us through the spatial constraint results for a single species first, before we zoom out and kind of look at the bigger picture. And I know there's a lot going on here, so I'll try to break it down for us. So this is our friend, the summer flounder again, on the Northeast shelf, both in the fall on the left side and in the spring on the right side. And both of these seasons are laid out exactly the same way. So we'll start by focusing in the fall on the left. So the series of map panels here are meant to help us visualize size-specific abundance and also spatial extent. And so these go from the smallest size group to the largest size group as we move kind of left to right and top to bottom. And the bar plot in the bottom middle row kind of shows us the components of constraint. So those consistency scores, extent scores, and constraint scores for each size group. And on the bottom right, we have this curve here, which shows us the relationship between spatial constraint and ontogeny. So if we stick with the fall and start at the very top left of that left figure, that's the larvae. And so the heat map underneath shows us abundance, but what we're really focused on are the little spread of red dots, which might be kind of hard to see. And so what the red dots do is they represent the core of the distribution that make up spatial extent. So we can see here that the core of the larval distribution is actually pretty large. They occupy kind of a lot of southern New England and trail into the mid-Atlantic bight. And the smallest juveniles, which is the next plot over, kind of the top middle of that fall side, those smallest juveniles have a core of distribution that's kind of near shore and focused in the southern region. And as we progress through size, the core of the distribution spreads both further north and further away from the coastline, kind of spreading out. But when we look at the final spatial constraint scores, the larvae actually end up being the most constrained because even though they're pretty widespread, they actually occupy that region really consistently. So the opposite is true for the spatial extent for the juveniles, where they're using a really small spatial area, but they use that area really inconsistently. So they end up with a low spatial constraint score. So we end up with a relationship on the bottom right figure there in the fall, where spatial constraint is on the Y and ontogeny from early to late life, from left to right on the x-axis. We end up with a high early life history spatial constraint that decreases through the small and middle sizes and then starts to tick up slightly again towards the largest sizes. So that's the fall. If we look at the spring, the larvae here on the top left are the same, it's the same data, but the size groups are all now shifted toward the shelf break. So in the fall, everybody's kind of near shore, and then in the spring, everybody's shifted out. But even though we have really distinct different uses of the shelf habitat, different areas in these two different seasons, the summer flounder actually end up having a really similar pattern of spatial constraint through ontogeny on those two bottom right figures. This suggests that summer flounder larvae may overall be the most spatially constrained at that larval stage and limited in where they need to be and where they can occur, while the other size groups in both seasons are less constrained and maybe more flexible in their spatial habitat use and maybe more able to respond to changes in climate. So that's the result for one species here. Next. This is what it looks like when we zoom out and look at all of our species together, or almost all of our species together. So these are the same spatial constraint versus ontogeny curves that we saw for summer flounder, but for all of our species on the West Coast on the left, and then for all of our species in the Northeast, just in the fall on the right. And this is super busy, but the only thing I want you to take away here is that there's a ton of individual variability. These patterns are going kind of all over the place. And the plots here are colored by broad functional groups, so elasmobranchs and flatfish and so on. And at first glance and with further analyses, there aren't any clear relationships and patterns of constraint through life among these species groups. Individual variability really does dominate here. Next slide, please. So taking all of that individual variability into consideration, we wanted to take ontogeny out of the picture just for a minute and ask whether there were species groups that were more or less constrained overall across life. So are there species that have stronger spatial habitat associations overall that may contribute to climate vulnerability? So to do this, we calculated a lifetime spatial constraint, so a mean constraint across life for each species. So each of the bars here is an individual species, and the height of the bar is that lifetime spatial constraint score. The bars are colored by their species type, and I removed the species names here just to make it a little easier to look at, a little less cluttered, and then they're sorted from high spatial constraint to low. And what jumps out at us first is that the flatfish in that kind of golden yellow color are the most constrained through life. And this might make sense because we know that flatfish, they associate really closely with bottom habitat, often directly with the bottom habitat. And they often have very particular sediment preferences, so particular sediment types, sediment grain sizes that might be limited in space. On the opposite end, we have the rockfish in kind of that dark purple color. And rockfish have the lowest overall lifetime spatial constraint. And this doesn't necessarily mean that rockfish don't have really strong habitat preferences. It just means that in this case, those preferences aren't necessarily geographically limiting. Next slide. So finally, what we wanted to do was ask whether there were species traits, and this is bringing ontogeny back into the picture. So looking beyond our broad functional groups and looking at whether there were traits that were associated with particular patterns in ontogenetic constraint. So to do this, we looked at the sensitivity attributes from the NOAA climate vulnerability assessments. And I know many of you are familiar with these climate vulnerability assessments. They've been conducted for fish stocks across many regions of the US, I think all regions at this point, including the Northeast in this paper on the top by Hare et al. In 2016. And then for the California current, which represents our West Coast region, in this paper that's on the bottom left by McClure et al. In 2023. And through this climate vulnerability assessment process, species were assigned scores for a suite of sensitivity attributes. And so these sensitivity attributes are meant to represent traits that reflect climate vulnerability. So things like adult mobility, habitat specialization, prey specificity, things along those lines. And these scores, as part of the climate vulnerability assessment process, were developed by a panel of experts. And so that blue box that I've highlighted on the right there shows an example of just one of these species scorecards for one species. I think it's winter flounder in the Northeast, where it's been scored for a series of sensitivity attributes. And those scores are all within that blue box. And then on the right there with those kind of stoplight colors, that's kind of the spread of the individual expert kind of opinions and scores that were assigned to each of those sensitivity attributes, ranging from attributes that were not very sensitive to climate to very sensitive to climate. Next slide. So we looked at these sensitivity attribute scores and compared them alongside our patterns of spatial constraints, so those constraint curves with ontogeny. And we treated those as two paired multivariate data sets. And we analyzed these together using a redundancy analysis, where the sensitivity attributes on the left were treated as the explanatory variables, and the spatial constraint patterns and those scores of constraint through ontogeny on the right were treated as our response variables. So really what we wanted to know was, do any of these sensitivity attributes explain the patterns of constraint through life? Next slide, please. And so what we found was that there was one attribute in particular, complexity of reproductive strategy, that contributed most to explaining patterns of constraint. So species with high reproductive complexity, according to the climate vulnerability assessment, are species that need things like really fine environmental cues in order for spawning to occur, or they have strong spawning site fidelity, things along those lines. Those are species that have complex reproductive strategies. So in the figure on the left, that's the results from our redundancy analysis. You can think of this basically like a PCA, or principal components analysis, where it's just two orthogonal axes that are kind of maximizing variation. And so the dots here are each individual species, and the color of the dot represents that reproductive complexity score. And so what we see in this figure is that many of the species with high reproductive complexity scores, so those yellows and those light greens, group together with similar patterns of ontogenetic spatial constraint. They have similar species curves. And to look at this a little bit further, we split our species into high and low reproductive complexity groups using a regression tree approach. And in the figure on the top right, the species with high reproductive complexity had this really distinct pattern of high early life history spatial constraint on that y-axis there that decreases pretty sharply through ontogeny on the x-axis. So species with complex reproductive strategies tend to produce early life history stages that are spatially constrained and that may be limited in their capacity to shift distributions. The bottom right figure there shows our low reproductive complexity group, and that group just had way more variability. So there was no distinct pattern of constraint through ontogeny there. Next slide. Okay, and so this here is just to show you a quick example of one of the species with our that's in our high reproductive complexity group. This is Atlantic cod in the Northeast. And cod, we know, are species that have some degree of spawning site fidelity, and they also form really large spawning aggregations. So they do have a complex reproductive strategy. And this figure is laid out the same as ones that you've seen before, but we're just going to focus on the top left where the larvae are. And the larvae are the most constrained life stage for cod, and they're constrained onto Georges Bank, where they may be vulnerable to climate change because they want to be in that location. And on the bottom right there, that species curve with ontogeny, they have that same pattern of high early life history spatial constraint that decreases through ontogeny. That's pretty characteristic of our high reproductive complexity group of species. Next slide, please. Okay, so I'm going to wrap up my portion here with a couple of summary points. So what we found was that ontogenetic spatial constraint varies like a ton by species. And this really just highlights the importance of examining individual spatial life stage vulnerabilities that may be unique to each species. We also saw really strong seasonal differences in habitat use through life, which might be important to consider when we think about the timing of climate impacts and where vulnerable life stages may be constrained. We also saw that reproductive strategy can limit and can potentially limit spatial climate variability, and that early life history stages may be spatially constrained and regularly use the same dispersal pathways that could be disrupted by climate change. And finally, a sensitivity attribute that I didn't mention in detail earlier, but that also came out as significant in our redundancy analyses, was sensitivity to ocean acidification. This was a smaller number of species, but these species tended to have small juveniles with high constraint. And this might suggest that those small juveniles that might be dependent on bivalves or other crunchy food sources might be spatially constrained to the location of their resources during early juvenile phases, and they may be vulnerable at that stage. And I think that wraps up my portion. So I'm going to go ahead and turn it back over to Jim now for the rest of the presentation. Thanks.
Thanks, Verena. Let me make sure. Okay. So that wraps up the bulk of what we're going to talk about today, but I wanted to share a few comments just to wrap things up. There is a couple elements of this project that are still ongoing. We are really hoping that they would be more complete to include with this because they're a pretty important component of this, but as it stands, there's still work to be done. And those things are, we are currently conducting life stage specific future projections of species distributions. So the motivation for this, a lot of previous future projections of species distributions are done, by aggregating all life stages together. And these approaches are really based on where and when these particular species are caught and what the environmental associations are with those catch locations. So it follows that we would see pretty different big differences amongst the life stages, especially for species like summer flounder, where we see pretty big spatial differences throughout ontogeny. And related to that, kind of a coupled output, we are going to be calculating climate change exposure across different life stages, both historic climate change exposure and projected future climate change exposure. This was already done at the species level by the NOAA's climate vulnerability assessments, but we're going to do it across life stages. And then to kind of wrap all of this up, these future projections and also the ontogenetic constraint analysis, We're going to kind of culminate everything into a risk table analysis using all these different research products. So that'll include things like lifetime spatial constraint and other outputs from that analysis, and other output from this future projection analysis. And so the goal of this would be to produce a product that might be readily usable by management. And just a few last words about potential management applications for our work. And all this is based on conversations that we've had with a lot of folks that work with NOAA or that are part of the Atlantic States or the the Atlantic States Marine Fishery Commission and some of the different marine fisheries councils. But the ecosystem and socioeconomic profiles, which was summarized in a paper last year by Shotwell and colleagues, this approach uses species metrics to inform species vulnerability and try to bring in ecosystem information to stock assessments. But we thought that our analyses could potentially inform some of those species metrics or some of the life history conceptual models that they use. Also, the Commission's management policy and science strategies. I don't have the rest of the name of that document, but basically their guiding document for adaptive climate change. We think our results could be helpful for some of their efforts to assess species climate vulnerability. And then fisheries ecosystem plans adopted by the different councils. I know they vary from one council to the next, but I think our results could be helpful with some of those efforts as well. And I think, yeah, so that's it. Appreciate y'all listening. And here's our team again, and happy to take any questions.
Wonderful. Thank you, Jim and Verena, for the great presentation and walking us through all of the things that you found so far. I'd invite other members of the research team who are also panelists to come on video if you'd like to participate in the question and answer round. We did get quite a few that came in throughout the webinar. So I'll just start with ones. I think Lorenzo actually answered a couple of them already. So those should be in the question and answer box and everyone can see answers to that. But for the ones that haven't been answered, I'll just start right at the top. So you mentioned that all the spatial data were collected from fishery independent methods, but were still catch data. If the above is correct, have you considered how inherent restrictions to fishing, for example, gears or equipment that's used, general limitations of field sampling, etc., might have affected your results? And have you tried running similar analyses using acoustic telemetry spatial data? Some flatfish actually move quite a lot in some regions, so I'm curious if the same results would hold using those data.
Okay. I guess I can take a stab at that. I think I may have misrepresented some of the words there. It is all fishery independent data. So there's no actual catch data there. It's all from long-term surveys. And while it is all from long-term surveys, there are kind of a number of different surveys that needed to be kind of incorporated together among each of these different regions. So we did make efforts to account for differences in gear, especially with changes. I mean, they're pretty long surveys, so especially with changes in gear that occurred through the length of the time series or changes in vessel, things like that. Those were all accounted for as part of our modeling efforts. So I think we did make an attempt to kind of even things out throughout all those data sets. The question about telemetry is really interesting because you can learn obviously a lot of individual information and group information from telemetry and from telemetry studies and by directly observing the movements of individuals. And it would be really interesting to compare what's seen with the movement of known size individuals and where they go and where they move. But unfortunately, this method here with the spatial constraint analysis is a super data hungry method. And so, so it does require these kind of big long-term data sets with length-specific ontogeny data in order to be able to run these analyses. So it's something that would not be able to be applied in the same way to telemetry data, which is obviously also super useful and super informative. I hope that helps.
Great, thank you. Next question. How did you assess pelagic species if the adult surveys are all bottom trawls?
You want that, Verena?
You could take a stab at it if you want. So there are a number of pelagic species that are caught in the bottom trawl surveys, and I do think in the Northeast that actually like the herring assessments are those are conducted using herring that are caught in the trawl survey data. So they do, it's not a perfect representation, but they do catch enough of them for it to count. And I think based on our species kind of filtering methods, there were pelagic species that were caught kind of incidentally in the trawl surveys, but we really focused on ones that were showing up very regularly and that had a good representation of information in those trawl surveys. I don't know if Jim has anything to add to that.
Nope, that's good.
Awesome. Are you aligning existing council and agency risk assessment products for the risk table analysis that you will do next?
I think that would be a good goal. I think once we finish the projections analysis and where we're getting towards the finish line of the projections analysis, we'll probably circle back with a number of different council folks and see if we can shape that in a way that's the most useful to them. Because the goal of the risk analysis would be to most effectively feed into management. So we'll definitely be getting back in touch with folks before that.
Sounds great. Next question. How did management actions influence species distributions and interpretation of spatial constraint? Overfished species that have been recovering would likely show a shift to areas where they had previously been high, where there had previously been high removal from fishing pressure.
Yeah, that's a good question. I don't know that, that didn't shape our analysis. I could see that potentially leading to bias, but yeah, I think that's something we probably should look into for some of these different species. If anybody has any other comments on that?
Any other thoughts from the research team? No worries if not. Awesome. So for the next question, have you applied spatial constraint analysis to any diadromous? I think I totally just butchered that. I'm so sorry. Diadromous species. Do you consider them to be species with complex reproductive strategies?
I mean, that would be a high level of constraint for species that are spawning in rivers or vice versa. And we did talk about trying to bring in more data from estuaries specifically, but yeah, it just became a question of complexity in the data and the modeling approaches versus just having tractable results. But yeah, there's no anadromous fish in our data set.
Yeah, I'll chime in to say that we were specifically trying to to avoid anadromous fish because while we know that they do have these really strong spatial constraints, the level of spatial data in the rivers and things like that that we would need to be able to show that effectively is just something that's not available. And for the same reason that Jim just mentioned with estuarine data and the idea of trying to incorporate estuarine data for estuarine dependent species was something that would be really great to be able to do, but the level of spatial information that's available between the offshore region and the estuarine regions is difficult to compare, I would say.
Sounds good. So a question about the plots. You might want to go back to some of those after I read them. But could you explain the dashed line in the plots of constraint versus ontogeny? Are they confidence intervals? It looked like a trend of more variability at the younger or older sizes.
I assume it's referring to this figure.
Yeah, or any of the ones that look kind of like this. But yeah, the dashed lines are confidence intervals in kind of how tight they fit that curve. And they are, yeah, and you're right that I think if you go back one, maybe, Jim. one where it starts out pretty tight and then spreads out toward the end. So yeah, it is that it kind of fits that curve and that line more closely towards those earlier life stages, and then towards the end, when they're at the adults, it's kind of less likely that it fits that line exactly. There's more of a chance that it could be petering out the other way as well. So there's just a little less confidence in those older sizes.
That's pretty common with GAM models at the data margins. There's greater uncertainty.
Great, thank you. Thank you for this talk. Would it be possible to integrate state-run surveys into these analyses? The trawl surveys run by the New England states, including Massachusetts, New Hampshire, and Maine, and others would be useful examples of dynamics within 12 nautical miles of the shore.
We came into this really hoping to use the state surveys, and we did spend a lot of time pulling a lot of that data together. And this kind of relates to one of the previous questions as well, is a trade-off in adding that level of complexity, including the state surveys. It's something that we're very interested in doing, and I think it could, for certain species, be joined with the federal surveys on the shelf for this approach. It might require a little bit more of a species by species approach and not able to apply it so broadly, but yeah, I think it's possible, and we are, as a group, we're very interested in linking the state run data sets, and especially in the big embayments, with the patterns that we see on the shelf. So maybe in future years we'll be doing that.
Great, thank you. We just had another one come in, another question. My sense is that especially recently, we have better larval than egg data in Ecomon, but did you consider including egg data in these analyses?
I'll leave that to you, Verena. I don't know what egg data is available in Ecomon.
Yeah, actually, we did not consider using egg data, and I think Lorenzo jumped off the call already, but I do think that egg data is something that he used in some of his subarctic species, but I can't remember why we didn't go down that path, but that might be interesting to see, especially if you can see kind of the egg data that traces kind of closer back to where those spatially constrained, to where those reproductive actions, that's a weird way to say it, might be happening, and where those reproductively complex life stages, where those things might be happening that could result in a constrained egg or larval stage or early life stage, because that would be closer to the event itself. But yeah, we didn't go down that path. And I'm actually, yeah, sorry, that's all I've got.
Great, thank you. That gets through our list of questions in the Q&A box. I did want to point out that there was, talking about how this could be applicable to many different regions, states, so on in the US. There was a question, or more of a comment, to circle back with the HMS management division as they manage smooth dogfish. And not necessarily something that like everyone needs to know about, but I did want to use this as an opportunity to say, please do reach back out to the research team members when we share this information and the webinar through our follow-up email. I'll include their contact information so that you can ask more questions, or also reach out if there's other connections that you saw or heard of during this webinar. And yeah, with that, unless there's anything else that the research team would like to add, we got through all of our questions, and have a great day, everybody.
Nope. I appreciate everybody tuning in, and yeah, thank you.
Yeah.
Thanks,
Thank
everybody.
you. Thank you all so much for joining us. All right. Bye, everyone.